Okay. I don't know if that's someone from us. Okay. So welcome to all of you, and welcome to those who are joining us online. So it's a pleasure to have Blaž here. Blaž Brence is a researcher at the Zusa Institute in Berlin. He is working on his PhD broadly in the topic of bioimaging, looking at imaging using different modalities and designing methods to analyze these images. Blaž did his bachelor's and master's from Slovenia, where he is originally from, before moving on to Germany. He has also spent some time at BAM, which is the Berlin Institute for Materials, prior to joining the Zusa Institute, which is called ZIP. So today he is going to talk to us about a project which is also a part of his PhD. And I think this focuses on microscopy images with some, I see Drosophila in there, which is kind of, the fruit fly, which is all pervasive in this field. So look forward to hearing you.
Yeah, thank you. Should be good too, right? Yeah. Okay. So yeah. First of all, thank you everybody for coming to my talk. As BJ already said, my name is Blaž. I work at ZIP, and today I want to give you a talk about, hope not too long, a talk about one of the projects I've been working on during my PhD, during my so far 3 years at ZIP. It's one of the projects that I do in parallel with the others, but so far I think I've been working on this for 1.5, 2 years. And it is titled fully automated quantification of synaptic localization in multichannel Drosophila photoreceptor microscopy data. And it does sound bad when you just read it like this or when you see it, but let's walk through this. Okay, first I wanted to give you a little bit of background about the research consortium I work in. So it's a pretty huge consortium. It's called Robust Circuit. And the main idea of the consortium is to study what is the role of noise in brain wiring. So noise is something that is traditionally associated with something faulty, something people want to avoid or neglect, so on. But there are some studies that actually show that noise can be actually beneficial for developing robust and well-working, functioning neuronal circuits. So within the project, we have 13 different laboratories from 5 different institutions. So most of us are in Berlin. There are some groups in Mainz and 1 in Paris. There are in total 8 neurobiological projects and 1 technical project, of which I'm a part of. And why are we actually part of neurobiological project is because 7 out of 8 of those projects actually require some kind of images to be taken to understand the development of neuronal circuits and so on. So we are here to actually develop the methods that help with the image analysis. Right. So first I want to give you a little bit of an introduction about what exactly we're doing. So you're familiar with brains. So brains are extremely complicated networks of neurons that are still they're already getting understood, but they're still far from being well understood. So for example, a typical human brain has 86 billion neurons and let's say 3,000 to 5,000 different neuron types. And all of these neurons are connecting to each other in a very specific manner. So they don't just connect to any neuron, they connect in a very specific place, location, time, with a specific type of neuron. So if we take a look at this neurons connecting with each other, so basically 2 neurons connecting are called a synapse. And if we take a look at the synapse formation, we can look at it from 2 different aspects. So first is, how does the synapse form? And this question has been researched a lot and is already everything is quite characterized. But there is the second part of this question, which is what controls where and with who with whom, well, with whom, I mean like with what neuron type does synapse form. And this is actually the part that we want to address within this project. So Drosophila Melanogaster is a common fruit fly, as you can see here, and for many years has been like a model organism for different research regarding neuroscience for multiple reasons. So one of them being the simplicity of the brain compared to human brain, because Drosophila brain contains only around 100,000 neurons, but despite having only 100,000 neurons, it still has 2,000 to 4,000 different neural types. So compared to the human, it has way less neurons, but it still has a really diverse array of neurons, of neuron types. Right, so this would be the Drosophila brain, and you can see this part is called the central brain, and on each side you can see we have an optic lobe, which is, well, as the name says, it's responsible for processing of optical signals, so basically for the fly to see. And each of those optic lobes contains 800 ommatidia. And if you look at this image, you can see, well, this is one optic lobe. And you can see there's like this kind of pattern here of, I don't know, like circular hexagonal structures. And these are actually the ommatidia. So there's 800 in each optic lobe. And if we take a look at this model of, well, this sketch of the figure of the optic lobe, you can see this is the retina. So basically this upper part is what you see here. And these small columnar structures are ommatidia. So each ommatidium contains 8 photoreceptors. They're called R1 to R8. And they extend from the retina. So from this ommatidia into the lamina, which is this part of the brain, and 6 types of them actually terminate here. So R1 to R6 terminate here, but 2 different types extend further into this part of the brain, which is called medulla. And yeah, R7 and R8 actually terminate here in different layers of medulla. As you can see, you have annotated different layers. So from M1 to M10. And depending on the neuron type, they terminate in different layers of medulla. So, like, how is all of this going to help us study the synapse formation, or how the neurons wire with each other? If we take a look at this work that was already done in 2020, so it shows us the R7 photoreceptors. So this layers, as I said, like M1 to M6. So they are located in medulla in this region. And if we take a look at this first picture, here we have the control group. So this means that there has been no genetic mutations to this animal. And you can see that the majority of but basically what we can see here is the cell, and the green dots are the synapses. And here we can see just the synapses. And you can see that the majority of these dots, the synapses, are actually located between layers M4 to M6. And this is what usually happens, because the synaptic partner of this cell, which is typically cell called DM8 or DM9, is located around here, which means, well, this cell is connecting to DM8 or DM9, and this is what we will expect, basically. But if we take a look at the other mutations, so the mutation ATG7, you can see that we have quite more synapses actually located in this upper part. So what does this mean? Because we know that DM8 or DM9 are located around here, and we can detect some synapses here. This means that this cell, in case of this mutation, must be connecting to another cell actually here. So our goal is basically to detect which types of mutation actually cause this behavior. So for the cells to connect with cells which are not their typical synaptic partners. Okay, so what exactly is the motivation for this work? So in the entire project, we have around 5,000 three-dimensional, three-channel images to analyze. And even if those were really simple images, analyzing 5,000 images is an insane task. So we want to prepare some kind of workflow that will do this automatically, or at least aid the analysis to the neuroscientist. So what kind of data we have? As I said, like, yeah, 3D, three-channel data of the fruit fly optic lobe obtained with confocal microscopy, and the voxel size is anisotropic. I don't know whether this is important, but for example, in each optic lobe, we should have around 1,600. So for example, in medulla part, we should have around 1,600 of neurons. And in case we see all of these neurons in image, we will not be able to distinguish anything. So we experimentally label a 2% subset of those photoreceptors in each image. So we end up with, well, 2% of those 1,600, around, let's say, 30. The 3D images or confocal stacks are of dimensions 124 by 124 times Z. So the Z varies, so between 280 and 350, 60. Images are acquired at, does it work? Images are acquired at 12-bit bit depth, and there's some 3D median filtering applied by the imaging software. So the three channels that I talked about, so the first channel would be the brain background. So from this channel, we can distinguish between different regions in the brain. So as I have mentioned, lamina, then we have medulla, and we have two other parts, which are lobula and lobula plate, which are, this one would be lobula, this would be medulla. But lobula plate and lamina are unfortunately not visible in this image. Then we have another channel, which is membrane marker. So this one actually tells us about the morphology of the neuron. By membrane, I mean the membrane of the neuron, of the photoreceptor. And you can see this is just one slice of the data. We can see some of the neurons here. And then we have another channel with synapses. And well, basically here we label the synapses. And on the first glance, this seems very similar to this, but if you take a closer look, you can see actually that it's kind of granular, and each one of those granules represents a synapse.
So what does labeling a synapse mean? Is it segmentation or is it?
Sorry, labeling of
the synapse?
So what I mean by labeling that the brain is treated so that a specific fluorophore is attached to the synapse, so that later when we image this optic lobe, the synapses are actually going to light up and they're going to be visible in the image.
But then how do you select only 2% of them to be?
So this gets very neurobiological, and it's not very understandable to me either, but like you treat the brain at the development with a heat shock of 15 minutes, and this actually causes only specific, some it's stochastic, so only specific amount of neurons are actually going to light up. So it doesn't mean that the other neurons die, but only specific subset of these neurons is going to light up during the imaging. We skipped one slide, yes. So now we come to the main part after the introduction, the workflow. As I said, we want to, the main thing is speed up the analysis. And the input to the workflow would be a repository, a folder with this 3-channel 3D images that the user wants to process. The images are processed one by one, so sequential. And the output of the workflow would be, the results, the readouts that the neurobiologists want. And I'm going to be talking about this later. And intermediate results, and by intermediate results, I mean just segmentations of different channels that the user can use to proofread the results. As the entire code actually grew quite quite big. There is multiple user-definable parameters to this workflow that one can adjust within the configuration JSON file. Right. So the first part of the workflow would be pre-processing. So we have this 4-dimensional image that we basically just split by channels. So we get 3-dimensional images, one for each channel. Then we do some image contrast enhancements. So as I said, like we acquired the image at 12-bit, and this is actually not necessary and causes, well, 12-bit data is saved as, well, 16-bit data, and it causes everything to just take so much more time. So we realized that actually 8-bit is just enough. So we downsampled to 8-bit, but we don't just rescale from 16 to 8-bit, but we actually rescale by the product of mean gray value and standard deviation. Because what can happen, it's not really well visible. This was supposed to be brain regions, but for example, here's some debris. There's something labeled that actually is undesired. We don't want this. And this part is super bright. And if we just basically rescale from 18-bit, from 16-bit to 8-bit, this part is still going to be very visible. But if we rescale by this product of mean gray value and standard deviation, it becomes a little bit dark, and then we get more contrast in the rest of the image. Then we have resampling. So as I said, like the voxel size is anisotropic. And we do some resampling for the channel 2 and 3, well, which are the channels with membrane and the synapses to isotropic voxel size. Now, if you ask me why we do this, just because I thought it's going to be simpler when I started this, but now after 2 years of developing it, I realized that this is not really necessary. But at this point, I have so much code written for isotropic images that, yeah, maybe I fix this at some point, but not for now. Then the next part of the workflow would be brain region segmentation. And here you can see like the typical slice of this brain region channel. So this will be, as I said, like medulla, lobula, plate, and lobula. How this is done, it's a multi-class segmentation, as we have 5 different classes. So lamina, then we have medulla, and we actually divide medulla into 2 parts. So peripheral medulla and the central medulla. So this one is a little bit more outer part and more inner part. Then we have lobula plate and lobula. Hence we have multi-class segmentation, and we use NNUnet for this. The segmentation is done in 2D, so in per slice in XY slices, as we have, well, the best resolution there, as this channel is not resampled, which means that, well, we segment each slice, and then when we have segmentation, we just stack those slices back up, and we get basically the 3D representation of the brain regions. There can be, because of this 2D approach, we can have, well, it's not visible here, but there are actually some artifacts. And so there is like some small part of the image which is labeled, but it shouldn't be. So we perform some artifact removal. Training was done with 20 manually or like semi-automatically annotated images within Amira software. The next part would be processing of the photoreceptors. So the photoreceptor channel. And this is actually done in 2 stages. First,
like, quick question. Just
yeah,
to
please.
Understand the segmentation in the previous
Sure.
slide. So these 5 regions. Is this the actual rendering of the brain as it is, or is this kind of an exported view?
You mean this one?
Yeah. So, for example, the peripheral medulla and the central medulla, are they actually adjacent to each other?
Yes, they are.
And the lamina is kind of.
Yeah, it's separated a little bit. Yeah. So it is rendering as it is. So this two.
And are there clear kind of boundaries between the peripheral medulla and the central medulla?
Yes and no. So basically, as I said, like medulla has different layers from M1 to M10. And peripheral, we have decided it's going to be from M1 to M4, and M5 to M10 are going to be the central medulla. You can see actually there's, again, it's not visible, but there's like kind of like a really faint white line going here, and this was kind of the border between these layers.
Some kind of a boundary. It's not just a.
It is the same region, but with this labeling, with this imaging technique, we can actually distinguish between them. But they stick to each other otherwise, while the others don't exactly. And next, back to the photoreceptor processing. We do it in 2 steps. First is the binary segmentation, and then the instance segmentation, because, well, we do need instances of photoreceptors. So first, the binary segmentation is done, again, using NNUNet, but this time in 3D configuration. So we take like small 3D patches from across the brain, and we run the prediction for each patch, and they will basically get stacked. So this part would be actually lamina, and this part down here, well, it's not visible because this is on our channel, but this would be medulla. And these are, well, again, it's not very well visible now, but these are of different intensities. So this part of the image looks quite different than this part of the image. So hence we decided, or we got better results actually training 2 different models, one for lamina and one for medulla. And then we basically use these 2 predictions. So automatic cropping is performed. So this crop box is estimated as the smallest box that would fit the entire brain region. Then this crop box is applied to this another channel, and well, the cropped image is used for prediction, and then the entire thing is fused. And here you can see the result of this binary prediction overlaid with the brain regions. So you can see, lamina and 2 parts of medulla. And you can see here, well, white lines of R1 to R6 photoreceptors, which terminate in this area, and R7 and R8 photoreceptors, which extend through lamina down into medulla. The training for this was done by manually tracing 10 images in Fiji. And then we have to get the instance segmentation. So how do we do that? If we take a look at this image, we see these long axons, well, cables kind of coming through, and these are actually not of our interest. So what is of our interest is the terminals of these photoreceptors, which are located inside of the brain regions. So to simplify things, we use this brain region channel as a mask, and we mask out everything that is not located within the brain regions. Then we perform the connected component analysis, and also, well, small components can be excluded, and this is what we end up with. And you can see different colors representing different photoreceptor instances: R1 to R6 in lamina, and R7 and R8 in medulla. Right, so I haven't been talking about this yet. There is another step to this photoreceptor processing. So the photoreceptors that are in medulla, R7 and R8, are actually of higher interest than the ones in R1 to R6. So for these photoreceptors, we don't only want to know, okay, what is the count of synapses here or something like this, but we actually want to know the distribution of synapses along these neurons, the photoreceptors. So in order to do this, actually, we need some kind of reference axis fit into these neurons. So we decided to fit in center lines, and well, these are linear lines. And if you remember, I've been talking about this ommatidia. Ommatidia, and then like the columnar structure. Because of this, columnar structure, it's okay. I mean, just the line is a good approximation. So we decided to fit in lines in each of those photoreceptors. So how do we do it? So for each photoreceptor, we extract a point cloud, which is 10 times downsampled, or just for performance reasons. And then we project these points for each photoreceptor into 2D. So into each point cloud of photoreceptor, we fit a best fitting plane. And this plane is determined by performing PCA to get the two components of PCA. Getting their vector product gives us the normal to the best fitting plane. And then we project these points into this plane. So basically points of each photoreceptor projected on the point on the plane that is fit into this point cloud. Then using Thielsen regression, we fit in a center line, and the points of the center line are going to be later projected back into 3D. So we end up with something like this. Yeah, so we have photoreceptors and then these lines, the center lines fitting them. Okay, as I said, we use the center lines for a reference. So basically, each photoreceptor is going to have like its own kind of reference cylinder coordinate system, and this is their axis. And we also need to figure out what is exactly the origin. So is the origin down here or is the origin up? So we figure out, I mean, we determine the origin by looking at the edge voxels of each center line. And we take the one that is closer to this peripheral medulla border. Yeah, so basically this is how we end up with the origins. And okay, now, I mean, it sounds good, but the center line fitting still didn't work sometimes well because actually these photoreceptors come into the medulla with an angle. So you can see they kind of grow with this angle, and then the angle changes and yeah, it starts growing in the other direction. So this part of the photoreceptor, we don't actually want to use this part for the determining what center line fits in. So in order to get rid of this and improve centerline fitting, we implemented two mechanisms. So the first one would be that we remove some percentage of the starting voxels of each point cloud. So we would remove, let's say, 10, 20% here. Or we can additionally shrink medulla. So this is the brain region here. We can additionally erode it and use it as a mask. So we would mask out this part. Or I don't know, depending on like how many voxels we shrink it by. And you can see, okay, this is the red one would be fitting without any of these mechanisms for fitting improvement, and the blue one would be after. So much better. Okay, the following step for the processing of the last channel of the synapse channel is done using another deep learning tool called Spotiflow, and it's a tool for spot detection in microscopy, in fluorescence microscopy, and it's again done in 2D by slice. And here you can see like this is one slice of this channel of like here you can see just a single photoreceptor, and here you can see the detected synapses. And in this case, we used a pre-trained model that was trained on some diverse microscopy data set. And as I said, like we do it per slice. So at the end, we stack up the slices again back together. But unfortunately, what happens is that in different slices, we can have the same synapse represented by multiple voxels that are like adjacent to each other in slices. So we make the assumption that the voxels in adjacent slices are in the neighborhood of each other. And we again run the connected component analysis. And here you can see, I don't know, like where, here you can see this kind of lines representing synapses. Each different color would be one synapse. But we want to get like just to a point cloud, like this, where one voxel represents one synapse. So how do we do this? We take the center of mass of n brightest voxels for each of those components. Yeah, so again, because we use the pre-trained model, it can happen that we actually pick up some noise that is actually not located even near to the neuron. And well, we don't want this in our analysis. So what we do is that we mask these predictions with photoreceptors, because we know that the synapses should be located within the photoreceptors. And here you can see some results. So this is volume rendering of this photoreceptor channel. And here you can see the predictions of synapses, and they correspond fairly well. And this is just an image of all the predicted synapses superimposed on the image of predicted photoreceptors.
So image, I didn't follow that. The lower right image, you said there was only a single point representing what.
So we want just each synapse, we just want it represented with a single voxel.
Okay.
Yeah, but because we do it by slices, so it means like if two slices, well, they're very similar adjacent slices. So probably the same synapse is going to be highlighted in both of those slices. So after we stack them back together, we're kind of going to end up with, well, with these lines representing synapses. I don't know whether this is clear. I know it's not very intuitive.
So just to clarify, so the right images are for slices, whereas the bottom.
Sorry. Yeah. So this is, well, both is actually volume rendering, but here you can see just slices stacked on top of each other. And the bottom one, we take, let's say, the average center of mass of for each.
How does that compare with the lower right image?
This one?
Yeah.
So for example, here you can see this dots, the voxels would be, each voxel would be one of those spheres rendering a synapse.
Okay. But then I see multiple of these
Here?
golden dots along one of these.
Yes. So I mean, yeah, maybe it's not. Now that I think about it, it's not very intuitive. So in this case, the photoreceptor is like laid horizontally, and this in this case is vertical. So if you would rotate it by 90 degrees, basically you would have the same thing. Okay. And in this case, we have representation for just one photoreceptor, and here is the rendering of all of them.
Okay.
Yeah. Okay, so now we came through processing of all of these channels, and we have this good segmentation of the brain regions, of the photoreceptors, and also the synapses. So now we actually have to make some meaningful biological readouts from this. So for each photoreceptors, as I said, like we have R1 to R8 photoreceptors, and for each photoreceptors of any type, we do the following readouts. So first would be the brain region. Well, this one is quite obvious. So if the photoreceptor is inside lamina region, we would it automatically gets assigned lamina, and if it's in medulla, it gets assigned medulla. Then we have the subtype. So as I said, like R1 to R6 are located inside lamina. So if the photoreceptor is in lamina, it's also automatically R1 to R6. And to clarify this, these are actually 6 different photoreceptor types, but with our experimental setup, they're indistinguishable. So we treat them as just one category. And for the photoreceptors in medulla, if they terminate in this central medulla, they are assigned R7. And if they terminate in this peripheral part of medulla, they are assigned R8. Yeah, so R7 terminates deeper than R8. Yeah, so for each photoreceptor, we also have the synapses assigned to it. So the synapse count and the volume in photoreceptor of the photoreceptor in voxels. Right, I didn't mention, and all the results are saved in JSON or and CSV format, configurable in the config file. But as I mentioned, the photoreceptors in medulla are of higher interest. So for those, we actually want to have some additional readouts. So what are those? The main thing is the synapse distribution, because the synapse distribution actually tells us where the synapses are along the center line that we fit. And this way we can figure out, if you remember my introductory slide, we can figure out whether another non-typical neuron type is actually binding or synapsing with this photoreceptor. So the user can specify the bin size of the synapse of this distribution. And well, we save the vector with bin distances, and then another vector which contains the number of synapses which correspond to each bin. And additionally, we save the distance for each synapse from the center line. So if you take a look at the center line here, okay, well, this is the photoreceptor, then we have the fixed center line, and we have the synapses. So for each synapse, we save how far is it from the origin here, and then we say also how far is it from the center line. And maybe this is not the best case for demonstrating the distance from the center line, but there are some neurons which actually grow in more L shape. Well, this one slightly, but more in L shape. So for example, it would extend here, and there would be synapses also around here. So in this case, the distance from the center line would be higher. Well, this is the most important readout. Then we have another important biological readout, which is distance of each photoreceptor from lobula and lobula plate. So here we have this, this figure that I've shown before, but yeah, and here we can see the brain regions. This would be lobula plate and lobula and medulla. And these two white lines, let's imagine that they represent photoreceptors. And for each photoreceptor, we calculate the distance from lobula and from the lobula plate. So for example, this is the posterior part of the optic lobe. So it means like in the back, and then anterior, the frontal part. And we know that there is developmental wave of these photoreceptors. So the photoreceptors that are in the back, so the posterior photoreceptors, develop faster than the one anterior. So by calculating the distance, we can actually figure out for each photoreceptor when compared to the other photoreceptors has this photoreceptor actually formed. So in this case, we would have smaller distance to the lobula and higher to lobula plate. And here would be exactly the opposite. So in this case, we would know that this one developed before this one. Another readout is the center line span in Z-axis. So because of the way that the brains were mounted and imaged, the majority of the center lines and the photoreceptors should lay within the XY plane. So in case we get a really high, Z-axis span, this would mean that it could mean different things, but like it could mean line span would be the reason to exclude this photoreceptor from further analysis. The center line. So along the center line, we calculate mean and median intensity for all of the channels. So one reason could be that this is quality criterion for center line feeding. And another reason could be that this way we can actually figure out layering of the medulla from the channel with brain background. Okay, now we came to the end of the workflow and we have all these results, but we still need to do some post-processing, and this is not part of the workflow. The workflow does contain some mechanisms for preventing saving bad results. So for example, the user can set what is the minimal size of each brain region, for instance. So if the brain region is segmented poorly and the size does not look good, this can be excluded. Or the user can set the minimum size of each photoreceptor, and those are going to be excluded. But for example, there's still a lot of things that can go wrong. So the responsibility for checking this is on the user. And if you remember from the previous slide, so for example, with this centerline span or like this median intensity or with the volume of the photoreceptor, the user can check whether these are actually biologically plausible. Additionally, the user can use this intermediate result. So in the software of her choice can check the brain region segmentation, photoreceptor instances, the central lines, or detected synapses. Okay, well, not very informative slide, but well, these are this is basically the CSV file that we get from the pipeline. Yeah, so there's one file with general information with the brain region, subtypes, and so on, and then we have another file with the profiles, so with its mean and median intensities, and then we have another file in which we save the synapse distributions. So now we come to the results. And well, firstly, about the technical results. The average running time for one image is approximately 56 minutes with the following setup. Run on the cluster. And how does this actually compare to the manual? With the manual, if we would analyze the image manually? Well, we are not completely sure, but if I estimate from the time that was necessary to create the training data, I would say that just creating training data for one image, and we're not talking about like actually mapping the synapses like to the layers of medulla and so on, it takes at least 10 hours. And probably if we're talking about, then writing down all the results and putting them in a form that will be understandable, probably double that. So yeah, huge improvement on this side, and we don't need anybody actually to be doing the work because it's done fully automatically. Then if we look at the results for the segmentation of brain region actually reached super good F1 score, and the binary segmentation of medulla and lamina a little bit lower, so 0.71 for medulla and 0.64 for lamina. And well, I don't know, maybe somebody could say that this is already getting low, but since from the network we get probability maps, we can also manually tune the threshold lower so that we get better predictions. And well, just by eyeballing it, we are very close to, well, basically getting all the neurons annotated.
So I forget, what was the size of the data set of the images for your training and tests?
So the training was done with 20 for brain regions, and for the photoreceptor binary prediction, 10 for each.
So is that like in this community, is that considered a big number, or?
Oh, well, I don't know. Hard to answer.
It's difficult to talk about the scores with the small data size.
Yeah, you're right. Well, for example, in case of brain regions, since we do the prediction in 2D, we don't actually have only 20 images, so we have 20 times the amount of slices. So I think like at the end we ended up with 10,000 slices or something pretty high. But in this case, yeah, we do have only 10 images, but the network also performs some data augmentation. Yeah, so
Yes.
So it does improve the quality, but yeah, I don't know. Like we saw satisfactory results with what we have done, so we didn't really attempt to to have more images.
So does it make sense to do testing on some phantom kind of synthetic region?
If somebody has available something similar, so there are some pre-trained models, but I didn't try them out, to be honest. So in my not first attempt, but one of the first attempts, it worked pretty well. So I didn't really bother trying it out. Yeah, unfortunately. And okay, this is about the technical results so far. So as I said, we have 5,000 images. We didn't manage to run them all yet, but for the sake of writing a paper, we have tested this workflow on the dataset of 50 randomly selected images, and these are the results we get. So these are the counts of the photoreceptor per image. So the average count of photoreceptor per image, and yeah, you can see R1 to R6, 64, R7, 13.6, R8, almost 10. And there's also invalid annotation. And what does this mean? This means that these photoreceptors were probably poorly segmented, or well, these were like maybe some small segmentation bits, and it was really impossible to distinguish where would this actually fit. So this is mostly for the case in medulla. So for example, if a small bit only lies in the central medulla, but not in the peripheral medulla, this doesn't make sense, because like the network, the neuron should be extending down to here. So if it only lies here at the bottom, this means that this was either poorly segmented or is noise. So we have 1.4 of such cases in an average image. And okay, these absolute numbers don't really tell us much, but if we take a look at the ratio, so we know that there's a ratio of R1 to R6 to R7 to R8. So since we have 6 and we have one of those, we expect the ratio of 6 to 1 to 1. And we actually get pretty close to this. We get to 6.6 to 1.4 to 1. And this deviation, yeah, multiple reasons. One of them could be, well, poor segmentation noise. And another one could be that there is some slight bias caused by how we actually provided this thermal shock, so that more of R7 photoreceptors could be annotated than R8. So this would not be the fault of the network, but this is still something to investigate.
But is the distribution of photoreceptors uniform across all of the regions? Is that the assumption, or is that?
No, we're not talking about the region. So for each optic lobe, we know that it should follow this this ratio. So because we have 8 of them in each of this ommatidium, and 6 of them are R1 to R6, which terminate here, and 2 of them continue down, so R7 and R8. So we know that the ratio should be 6 of those to 1 of R7 to 1 of R8. Yeah. Okay, a couple more results. So what we have observed that we have large variance in volume, and this is, yeah, biologically does not make great sense, but we have explanation for it. So as I said, this is the data set with 50 randomly selected images. And one of the reasons could be, as I have mentioned in post-processing, at the post-processing slides, the responsibility to exclude poorly segmented neurons lays on the user. So this was not done in this case. So as we use connected component analysis, it could be that if 2 binary predictions touch, they would be labeled as a single neurons. So from the volume readout, one could exclude them, but this was not done in this case. Then we have the synapse count for different photoreceptor types, and well, this is the synapse count of biologically plausible range. So you can see that, well, some synapses are actually detected in biologically implausible range. And well, our explanation for this is that we used a pre-trained model because when we first started working with this SpottyFlow tool, it was not actually available in 3D. So we used the one in 2D. We didn't create the training data. Well, long story short, the synapse detection is still something that is work in progress. But okay, while these numbers are actually a little bit lower to what is reported in prior literature, if we take a look at the distribution, so distribution of synapses for R7 and R8, you can see that it does actually follow this distribution that is expected. So the majority of synapses for R7, which terminates deeper in the brain, are located between, let's say, 15 and 25 micrometers, and R8, which terminates a bit more shallow, they're detected between 5 and let's say 15, 18 micrometers. So this does biologically make sense. And yeah, this is another interesting readout. And here we have the plot of photoreceptor distances from lobula and lobula plate. So if we take a look at this edge case, you can see this is the photoreceptor, and I believe this would be the posterior side. You can see low distance to lobula and high distance to lobula plate. And if we look at the other extreme, we have high distance to lobula and low distance to lobula plate. So basically this way we can also see that, well, this individual photoreceptor has developed before this photoreceptor. Okay, now if I come to the conclusion about the workflow, it is, well, it's, I don't know, I wouldn't call it fast considering it still takes an hour to run, but it is absolutely faster and it's objective. So this is also an issue in, I guess, in multiple different fields, but and also in one of them being neurobiology, and that in case of high amount of data that we want to analyze, maybe multiple people will actually work on it. And this introduces a lot of subjectivity. So what might seem like a synapse to one person or a well-fit center line to one person does not seem to the other person. So this is an issue that actually we solve with workflow because it's objective. So the approach of actually mapping synapses in this comparable centerline based coordinate systems, this is something novel that hasn't been priorly done. What was done before is, well, basically manually, not annotating, but manually checking for each synapse in which layer it actually belongs. And this is, yeah, very time consuming. No, okay, now I talked about all the amazing things about the workflow, but well, there are some drawbacks. So the workflow that we have created is highly customized for our data. So I mean, the entire issue is highly specific, and the workflow in its current state is not applicable to different type of data. But it is written in a modular fashion. So a lot of parts can be actually repurposed for other work. So there is another project I'm helping in, and where we also have kind of columnar-like cells, and these are called TM2 or TM9, and they're trying to do actually very similar analysis. So with this modular workflow, I can repurpose some parts of this workflow for their analysis as well. Of course, I will probably have to train a different model for photoreceptor, in this case, the neuron prediction. Yeah. And the workflow was submitted in the form of short paper to BCBM just last Friday. And about the future work, so I think the majority of stuff is already done, but we still need to do some improvements. So the first one would be we want to improve the synapse detection. And as I said, first, the spot flow was not available in 3D, but now it is. So now we have created our own annotated data, and we're already working on the 3D model, which hopefully performs better. As I said, neuron segmentation is done with connected components, and it works fairly well for, I don't know, like eyeballing 95% of neurons. But this other 5% of neurons, for example, is, well, two neurons touching, and they get annotated as a single neuron. So by implementing something a little bit more sophisticated, like watershed segmentation, hierarchical watershed segmentation, we could actually split them. There is still another thing that we want to work on, but we didn't manage, or we didn't have great ideas, is that we want to describe some spatial properties of photoreceptors. So we have thought of tortuosity, and this is something we have implemented, but maybe there is something better, and maybe, I don't know, this could be even a topological problem. So if somebody has some good suggestion about it, I'm all ears. There's a plan to write a full paper with neurobiological findings as well. And we have this, well, not yet, but soon we will have this huge amount of data that probably no one else has. So we try to figure out what else can we do with this. So one idea was whether we can perform some kind of dimension reduction and get the clustering depending on the neuron subtypes. So I don't know, might R7, R8 neurons might cluster in separate clusters. So, and I believe this should be it. Hope it wasn't too long. I can't really see a little bit. Yeah, well, thank you.
Okay, thanks. The online.
Where?
Could you prompt the people
Sure.
to join online?
Also, people online, if somebody has any questions, please feel free to ask in the chat.
I think they can speak out also.
Oh, really?
We'll be able to hear it.
Okay.
Am I audible?
I can
Hello?
hear.
Okay. So I have one question. None of the models are pre-trained, right?
Come again, please.
None of the model that you have used are
Yes.
pre-trained.
No.
Okay.
Yeah. Yeah, straightforward.
I think that's a feature of this. And then
Yeah, it is one of the features that you can use a pre-trained model. But well, since as I have said, it worked well straight out of the box with our data, I didn't bother trying it out.
maybe you have questions here. Any questions? Maybe I'll start off with some. So I have a couple of questions about the workflow. Right?
Okay.
So probably a straightforward question. So is this entire workflow available as just one package for the neurobiologists?
It is. Well, so it's not really available yet as it's work in progress. So it is available to my project partner, so we can do the testing and development. But yeah, it is. So at the very end, the user can clone the GitHub repo and just run the main function with Python function name and then the config file. Yeah.
And then out comes all the properties.
Exactly. So in the config file, the user can specify the input folder, the output folder for the intermediate results, and also for results in CSV or JSON file.
Okay.
So basically, the analysis runs for the images in the input folder, and everything is saved automatically in the output.
That's cool. So the second question is a little bit more on kind of user interaction, bringing the user into the loop. So let's say, so you talked about, for example, is it possible to allow for user input to nudge that those invalid configurations also into let's say R1, R2, right? You had a few in one of the charts that you showed, right? I think there were a bunch which were invalid, right? I think some of those in the previous slide, I think.
Here.
Yeah, those that 1.4 percentage, right? So is it possible to provide user input where they could probably see the segmentation, see the photoreceptor, and then fix the issue so that you can nudge that also? Is the workflow amenable to such user inputs?
Well, in the current form, no. I mean, we didn't even attempt doing something like this because the entire idea is to just let it run as it is, like, so that we don't have to actually do some manual work with it. Right. Because it's not feasible. But also, this is not really a problem because we have for each animal, like for each gene, we have 2 to 3 different animals imaged, and each animal has 2 optic lobes, which means we basically end up with, let's say, 5, like on average 5 image for each genetic mutation. And if 1 or 2 of the neurons are poorly segmented, it's not an issue because in each image we have still 30 or like let's say 15 of neurons of the same subtype that we can use for analysis. So yeah, these would be simply discredited.
Okay. But would the neurobiologists be interested in seeing kind of some of the intermediate results to gain confidence maybe not the ones we are already working with. Maybe they have seen the entire workflow, but let's say this.
I mean, so after running the workflow, you don't only see the results, but also all the segmentations are saved. So, I mean, they are being saved through the entire workflow. And then, well, depending on what user decides in the config file, they can be either deleted or zipped or just left as is, as it is. And then those can be loaded into, well, any software for image viewing to actually check whether results make sense. Yeah, to actually to get confident with the results. But it's something done after the processing.
Sure.
Not during. Yeah.
For the questions, let's thank Blaž again.
Thank you.
Here's something for to remember and see.
Thank you very much. Very much appreciated.
Right. Thank you. And he's around for the week, so in case some of you
sure.
You have questions, you could maybe come and introduce yourself now and then.
There's also my email if somebody
needs
it.
Okay. Okay. Thank you.
Thank you.
Thank you. Thanks, Blaž. Thanks everyone for coming.
Right. I don't know. I didn't check. Maybe.
No, but they had no further questions, so I think that would be.